Okay, well, thank you for joining today. This is going to be our first summer series talk, and today we're going to be having Calvin give his master's presentation thesis. But a little background about him. So he graduated from Cal State Long Beach in 2010 with his bachelor's degree in marine biology, and from Cal State University of Northridge in 2018 with his master's in biology with the concentration of ichthyology. Before joining Endemic Environmental Services, he worked in various marine-related positions. He was a museum collection assistant at the Cabrillo Marine Aquarium for 11 years, where he maintained the aquarium's historic reference specimens collections and was involved in various field research projects. Before grad school, he worked as a scientific aide for the California Recreational Fisheries Surveys through the California Department of Fish and Wildlife for 2 years, where he collected biological, geospatial, and economic data on various modes of recreation fishing. After grad school, he worked for 2 years as a project species observer for BioWaves Inc., where he monitored marine mammals and green sea turtles activity in and around an immunization pier project at the Seal Beach Naval Weapons Station, as well as within the Seal Beach National Wildlife Refuge. So with that, I will give the floor to Calvin.
Hi everyone, thank you for showing up and tuning in to my talk. I'm glad you're interested in actually hearing what I have to say. Okay, so I'm going to go ahead and share my presentation screen with you all, and you guys. Okay, so I just clicked on share screen, and it says host disabled participant screen sharing.
Oh.
So Anita, I'm not sure what that means.
Okay, hold on.
Okay. All right. Okay, can everybody see that?
Yes.
Yes. Let me know, speak up now if you cannot see it. Can everybody still see me as well?
Yes, we can see you. We can see the fish, whatever fish this is, sorry.
Yes, so this is a barred sand bass, okay. All right, so this work is my master's thesis project, and I kind of fine-tuned this talk for this particular presentation. So it's not going to have absolutely everything that I did because there was a lot of other things besides this, but some of the most compelling data is the stuff that I'm going to share with you today. Okay, so oh, I don't know why this isn't working. Okay, there we go. All right, so I'm going to start off by talking a little bit about some concepts first before we get into the work that I actually did. And I'm going to start off by talking about spawning aggregations. So an aggregation, loosely defined, is a gathering of fish in a common place at a common time and for a common purpose. And this is typically thought of as being different from a school of fish. It's very common for people to think that a school and an aggregation are the same thing, but they're distinctly different because a school is actually a discrete social unit. The fish will actually live together and move together. An aggregation is typically fish that are primarily solitary in nature and will only aggregate in certain times, in certain places, such as spawning. Okay, so spawning aggregation is the running definition that we have is that it's a gathering of fish in reproductively active condition. Even though the actual occurrence of reproduction can be somewhat variable. They don't necessarily have to reproduce, but they're in reproductively active condition, and that's the only thing that we can be sure of for a spawning aggregation. So obviously, when you have a lot of fish occurring in one area, and fishermen happen to know where they are, there's actually what we call an aggregation-based fishery, to where the goal in maximizing your productivity in your fishery is to maximize your yield while minimizing your effort spent fishing. And so obviously this is important for fishery type species. So obviously they all have to be there at the same time. These aggregations have to occur at certain times of the year or certain times of the day even in order to synchronize their reproductive activity. So that makes logical sense, right? So a good example are these Nassau grouper right here, where they typically spawn almost perfectly lined up to lunar phase. It's usually right around 8 days following a full moon phase is when they would spawn. So in addition to being there at the same time and synchronizing the reproductive activity, there is also some thought that they would aggregate during ideal times to maximize their offspring survival, and even as far as dispersal to ideal juvenile areas, because certain times of year, the currents will be running in certain ways, and this current could potentially promote transport to juvenile areas. So this is actually an idea proposed by Phil LaBelle, who did a lot of work on tropical damselfishes and butterfly fishes, and have actually found that the tidal currents during the times when they're spawning can actually promote this particular transport. So unfortunately, reproductive timing can be somewhat predictable among the fishing community, and so these spawning aggregations being predictable can potentially promote overharvest. Okay, so resource management is usually tasked with estimating the standing stock biomass or the health of the population of certain fishery species. And so what they would typically use is a measure called catch per unit of effort, or CPUE. So this is what we actually call fishery dependent data, because they're actually using catch counts from both commercial and recreational fishing data. So this is data that depends upon a fishery, okay? So there are two components to catch per unit of effort. You've got the catch, which is some sort of measurable yield, whether that would be total number of fish, which is usually, which is common in recreational fisheries, or the total weight of fish, which is common more in the commercial fishing realm. And the second component is you have the effort, which is some measurable or quantifiable work in order to obtain this particular catch. So that can be anything from distance traveled to the length of time actively fishing, also known as wet gear hours. And in the sport fishing community, it can be number of anglers on a boat would be considered effort data as well. So the way that they typically use this information is, very straightforward. If they have a high catch, low effort scenario, the interpretation is that the fishery is doing well, the population is doing well. And of course, in contrast, if you have a low catch and a high effort scenario, then the interpretation is that the fishery is not doing so well. So that seems to make logical sense, right? But unfortunately, there is a dynamic known as hyperstability or hyperstable fishery, and that describes an overestimation of abundance. And the reason why that occurs, it's because catch per unit effort, when used in a very simple way, tends to remain constant while the true population goes unnoticeably in decline. There's a couple of reasons for that. So catch per unit effort, unfortunately, comes with a couple of inherent sort of flawed, I guess, assumptions. The first being that catch per unit of effort assumes that fish are somehow uniformly distributed to where you can take a random sample of a particular group of fish, and that would be referred to as your estimation of or your representation of the health of the population. But we know with aggregation-based fisheries that that's not necessarily true. Fish are spatially variable. They can occur in the thousands in one particular area and be completely non-existent half a mile away, okay? Another assumption that CPUE makes is that fishermen are randomly sampled, which is also not true, right? Fishing efforts are usually targeted around areas and times when they know that they're going to maximize their production. So it's very far from random. They know where they are, they know when they're going to be there. And so you end up with a high catch, low effort scenario. Aggregation-based fisheries can be particularly susceptible to this because the overall population decline can be masked by a so-called illusion of plenty. Okay, so I'm going to switch gears here and talk about some abiotic factors. Well, one abiotic factor in particular that I looked at several, but the most compelling data that I had focused on lunar phase. So when we think about the concept of reproductive timing, there's a lot of environmental factors that cue for the timing of these reproductive events. And lunar phase in particular has been experienced among the fishing community for centuries. It's been known for a long time that the fish behavior is centered around lunar phase, or potentially can be centered around patterns in lunar phase. Even to the point where the lunar effects on fish behavior is actually fairly well documented in the scientific community. But the one problem, though, is that a lot of these previous studies focused on fishery dependent data. So of course, you're going to end up having some fishery bias because fishing efforts are often centered around times, again, of maximum yield. And so they could actually be targeted around certain lunar phases that they know they're going to be able to catch a lot of fish. Okay, so we're going to talk about the barred sand bass, which is my study species. This is an aggregation-based spawner, right, or an aggregative spawner. And they spawn during June, July, and August of normal years, so during the summertime when the water is at its warmest. So they're under family Serenity. They're actually one of 3 species in the Paralabrax genus that you can commonly find in California. They can reach about just over ½ meter in total length, and they can weigh up to 6 kilograms. So right around 12, 13 pounds, right? They're pretty widely distributed latitudinally. They can be as far north as Santa Cruz, California, to as far south as Mag Bay in Baja, California, but they're most commonly found south of Point Conception, so this little area that we refer to as the Southern California Bight. They comprise a really important recreational fishery, and I emphasize recreational because they're actually a recreational only fishery and have been that way since 1953. So whenever you talk to a fisherman and they think that commercial fishing is what's to blame and not recreational fishing, it's not true for anything in the Paralabrax genus in California because they're recreational only. Commercial fisheries are not allowed to target them at all. So within that same decade, a catch regulation was implemented in order to control how many fish people were taking. So they have a bag limit of 10 fish per day and a minimum length of 12 inches total length. They have to be at least 12 inches in order for you to keep them. This actually changed. So this remains the regulation for over half a century, but because of a very recent shocking decline, which I'll go over in a minute, the regulation actually changed back in 2013, where they cut the bag limit in half. You're only allowed to take 5 per day, and the minimum length is now 14 inches. And the idea behind that is to give them a couple of extra years to spawn to try to keep the populations up. Okay, so let's take a look at the historic catch counts. This is raw catch from 1963 to 2003. It's actually a little bit past that. It's actually just over 2010 is what this graph shows. And this is actual reported catch from sport fishing community. And you can see that it actually increased very steadily, and it peaked at just before the turn of the century, around in the mid-90s, at just over 2 million fish caught annually, and started to decline. But at the turn of the century, you would see a massive decline down from about 1.4 million fish caught annually to a mere 100,000 fish caught annually. So this was over a 90% decline in annual catch just within a 10-year span. So this is something that obviously had to be responded to very quickly. Now let's take a look at the CPUE, or catch per unit of effort data of that same time period. So the effort that they used was the number of anglers per trip. And so you can actually see that there was a spike in the early 70s of catch per unit of effort. And this is most likely, so this right here, once again, this is a high catch, low effort scenario, right? And this is most likely when these spawning aggregations were actually first being discovered, where they figured out that you can get a high catch with relatively low effort in these areas. So this is most likely where this spike came from. And then you can see it actually remains relatively stable across time. It doesn't show this increase. It remains stable even across when it starts to decline. You actually don't see that. It's not until about 2005 or so when the catch per unit of effort actually began to decrease too in response to this massive decline. So this is actually sort of a telling tale here. One of the analogies that my advisor had mentioned before that kind of stuck with me is let's say you've got a bucket full of jelly beans, right? And every day you close your eyes and you stick your hand in and you pull up a handful of jelly beans. This is catch per unit effort in the most simple sense, is that every day you're using the same amount of effort and you're pulling up the same amount of catch. Now, what's happening to your actual population? Your actual population of your jelly beans, right, in that bucket is slowly going down, but your catch per unit effort is still showing, oh, this is a really good population. It's still healthy, it's still stable, but it's not. It's slowly going down. It's not until it gets to a point where you have a hard time picking up a full handful and you're starting to scrape them out from the corners of the bucket that you actually notice, oh, hey, I think there's a problem. But at that point, the population of your jelly beans may have reached critical levels already. So this is what we believe is potentially happening to the barred sand bass fishery. Okay, so my research objective, obviously, I've set the stage where I'm not a huge fan of fishery dependent data. So one of the things that I looked for was fishery independent data. I was interested in looking at their spatial and temporal aggregation patterns, where they actually are in time and space, irrespective of what the fisheries are telling us. So I focus on lunar periodicity. I actually looked at several different factors, but lunar periodicity is the one that seemed to be the most interesting. So I thought I'd share that with you today. And I looked at variations in their horizontal distributions across a horizontal plane, as well as their vertical distributions in the water column, how high up they were, how far down they were in the water column. And that's important, and I'll go over that in just a minute. So I hypothesized that there would be significant spatial patterns in their spawning aggregations that would be associated in some way to lunar phase. Okay, so I looked at depth, their vertical distributions for a specific reason. When you look at their congeners, or the other Paralabrax species that you can find in California that I mentioned earlier, this is Paralabrax maculata fasciatus, the spotted sand bass. And the spawning behavior was actually described back in 2006. And what you see is a single female being chased by several males, and they compete for proximity to the female, and they will do a vertical spawning rush and synchronize their gamete release. So the female will release eggs into a cloud of milk, and males will release sperm at the same time, and they'll externally fertilize in the water column. And the reason why they go up, it's to put their gametes high into the water column to maximize the fertilization and to allow them to then disperse and spread out and drift along on the currents. So you can see a similar thing with Paralabrax clathratus. This is the kelp bass, or the fishermen refer to it as calico bass, and they exhibit the same sort of behavior or similar behavior, where a single female is rushed by several males, and they will rise up into the water column and synchronize their gamete release as well. So shown here is actually some barred sand bass data. This is from McKinsey et al. 2014. Okay, and you can see, now you may be asking why we didn't do this same thing for barred sand bass. Well, I'll go over that in a minute, but it's actually quite a bit more challenging. But what they did in 2014 was they implanted acoustic transmitters. They surgically implanted them into reproductively mature barred sand bass, and some of these transmitters actually had depth sensors, and they were able to track their depth activity over time. So you can see on this upper panel here, this is what they look like during the non-spawning season. They're bottom associated fish typically. Barred sand bass likes to hang out on the bottom. So you actually see they don't really rise up more than a meter or two off the bottom. But then during the spawning season, particularly during the morning to early afternoon periods, they suddenly will increase their vertical space usage right here. And so you can see that repeated bouncing up and down. They actually saw that they were on the surface and bounced downward and then back up repeatedly. And then even during the spawning season, during the off hours, right, during the later parts of the day and into the evening, they once again will return to the bottom to rest. And that's what you see in this third panel here. So why didn't I actually go in there to physically see them? Well, I actually tried. I tried very hard to generate an ethogram just like these two studies for the barred sand bass, but unfortunately they spawn over sandy low relief environments in the mid to upper water column where the water looks like pea soup. So I wasn't actually able to do anything visual for that reason. They're quite a bit harder to study for that reason. So instead, I used sonar. This was basically what I consider a camera that uses sound waves instead of light waves. So basically what a sonar does is it emits an acoustic signal into the water. That signal will then bounce off of objects in the water, and the transducer will then record the resultant echo. And you can do a lot of things with that. So the unit that I used was a Biosonics DTX portable echo sounder using a split beam configuration. Split beam basically means that the detection is actually sort of cone shaped. So it spreads out into sort of like a spotlight, once again, using sound instead of light, okay? The transducer that I used was a 206 kilohertz transducer. That's something that's, you know, sonar gurus, that's something that they know more about, okay? The cool thing about this unit is that it will continuously record throughout your entire cruise track. So all you would have to do is turn the unit on and then drive wherever you want to go. And this unit is just going to record acoustic information for your entire cruise track. And it's actually GPS integrated. And so you can synchronize GPS onto any part of your cruise track. All you'd have to do is just drag this line back and forth, and it will tell you exactly where you are in time and space. So this is what the unit looks like. It looks a little bit small just because it's a small picture, but this right here is a Panasonic Toughbook laptop computer. This is where all the information is stored. It is attached to a receiver box, and that is then attached to a 20-foot long deck cable that is attached to the transducer. This is the part that goes into the water. It was about a 15-pound transducer, so about the size of a small pumpkin. It was about that big. So you may be wondering what this is down here, this blue apparatus here. This is what's called a dead weight towing body. Now, I was using this on a small boat. It was a 17-foot Boston whaler. It was a flat bottom whaler that actually will move a lot on the top of the water. So if you were to look at the echogram here, you'll see all this movement on the bottom here. This is actually not representative of what the bottom looked like. This is actually recorded movement from the boat. And so to try to minimize that movement, I used a dead weight towing body right here, which is basically, it kind of looks like the mix between a snow sled and an aircraft. But the idea behind it is you mount the transducer right down there to the device, and you hang it over the side of the boat. And so as the boat's bouncing up and down with the waves, this transducer will not move in that same way, okay? So, of course, my analyses were performed on a software program known as EchoView, and I used version 7.1. I think they're up to version 14 now, so this is very outdated. Okay, so I'm going to show you here is a little video clip. Back in 2010, when my advisor first acquired this unit, this happened before I started grad school, he wanted to try to relate some actual video footage to what scenes look like on an echogram. And so this is actually the result of that effort. So what you're going to see are some northern anchovy being attacked by some barred sand bass. So here are the anchovy, okay? And you're going to see a barred sand bass appear right about there. Okay, boom, there it is. Okay, so these are piscivorous, they're predatory fish. Okay, so this was basically a scene captured on video, and the echogram looks like this of that same scene. Okay, so what you see here, these are the northern anchovy, and these right here are the barred sand bass. So it's important to note that it's a little bit tricky to look at an echogram because we're very tempted to think that this right here is a physical distance, and you're wondering, well, why are they so far apart if they were actually interacting with each other? So this right here is actually a difference in time because the unit is actively pinging, continuously recording. So if you were to stand in one spot and you're constantly metering a particular object, it's actually going to look like a solid line, okay? So that's something to note right here, that these anchovy and these sand bass were actually swimming in and out of the cone of detection of this unit. And it first showed the anchovy here, and then the anchovy may have swam away from the unit briefly, and then the sand bass moved in, chasing these anchovy in the same general area. So these were actually individuals that were right on top of each other, even though they appear far away on an echogram, okay? So it's a little bit tricky to look at it in that way. So just kind of showing off a little bit more of what that technology can do. So the echograms appear to be two-dimensional, but the unit is actually a cone of detection. So it actually collects three-dimensional data. And you can go into any of the targets and the objects that you see within the sonar, and you can actually do a three-dimensional visualization of these objects. So this right here would be sort of an example of some individual barred sand bass and a small aggregation that was captured with the sonar. Okay, so shown here is sort of this is a screenshot of the EchoView analysis platform. So you got to the left, you've got all kinds of cool information here, including the GPS integration and the target strength and all that stuff over on this left side. And then right here in the middle, you've got your echogram. And right here on the right, you've got your cruise track. Once again, it's GPS integrated. So there's a little crosshair here, and I've put a little red circle around the center of the crosshair to kind of orient you to where it is. And if you notice this line, this vertical line right here, this is actually a ping analysis line where I'd be able to, in the software, I'd be able to take this line and drag it anywhere I wanted to put it. And this map, well, this crosshair right here would actually show me exactly where I am along that cruise track, okay? So you've got some individual barred sand bass detections right here in the center. And what you'd be able to do is zoom in on these individual targets, and you'll actually see that they're composed of vertical columns of pixels that are parallel. Each column represents a single ping off the sonar. I had the sonar running at 5 pings per second, so it took about 2 seconds for it to capture this particular image. You can then trace a polygon around your targets and be able to generate a target strength that is contained within this polygon here. Basically what you're seeing here is a relative frequency histogram that shows the frequency of the different target strength values that are contained within this polygon here. Target strength is basically a measure of signal loss. It's always measured in negative decibels, because once again, you imagine a sonar is pulsing a sound into the water, and that sound scatters, right? It actually, some of it gets lost and dissipates. Some of it will strike the object and scatter off. Some of it will get absorbed, and some of it will bounce back. Okay, so there's a lot of signal loss based on the physical characteristics of that object. And so the resultant echo, what this unit is measuring is how much signal loss, and that it can actually be used as sort of an acoustic signature of certain species of fish. So a northern anchovy is going to absorb more signal than, let's say, something bigger like a barred sand bass, okay? So then how do you figure out the target strength of a barred sand bass? So my advisor back in 2010 had figured this out already. He did what's called an in situ target strength characterization, which is basically a fancy way of saying we caught a bunch of reproductively active fish and hung them directly beneath the sonar transducer and metered them directly. My advisor is very good at doing things in a very simple and logical way. Okay, so that's exactly what they did here. And this is the resultant echogram of that methodology. So what you're seeing are 3 barred sand bass and a 1 kilogram lead weight being actively pinged continuously. And once again, you can see this, this vertical line. And this is actually a line that's overlaid over a single ping. And what you can do is a target strength depth profile of that single ping, and you can see where all these peaks are. So it starts off at a really low target strength, and then it goes up as it measures the barred sand bass, of course, also the 1 kilogram weight, and then it goes back down. So you can see these peaks here are actually between -30 to -40 decibels of target strength. And it turns out that they average right around -35 decibels and a range of between -30 to -40. So now we have our barred sand bass acoustic signature. Okay, so a little bit about the study site here. This is actually a map of the Southern California bight, indicated by the black dots are the 5 known barred sand bass spawning aggregation sites, the largest of which being the Huntington Flats, which is about 3 to 5 kilometers off of Huntington Beach, California. The reason why I chose this site, number one, is proximity. I live the closest to it, but also because it is the largest site. So back in 2004 to 2008, here's some catch counts of the the summer months that was collected by Department of Fish and Wildlife. And you can actually see that nearly half of all barred sand bass caught during the summer months during 2004 to 2008 were actually caught off the Huntington Flats. So a little bit more details about this site here. So just to kind of orient you, this is the Huntington Pier right here. You got Bolsa Chica, Seal Beach Naval Weapons Station. Then of course you have the Port of Los Angeles up here off this map. And these breakwalls are actually a very important structure for barred sand bass. During the non-spawning season, they prefer ecotone environments, which is basically the interface between two differing habitat types. So like the Long Beach breakwall, for instance, is an ecotone. It's rock and follows abruptly to sand. Okay, so this is an ecotone environment that they would live on most of the year, except during June, July, and August. They'll actually travel out to this low relief sand flat environment known as the Huntington Flats. Okay, it's actually bordered to the northwest by the Bolsa Chica artificial reef. It's also known in the sport fishing community as Iser's Reef. It's named after someone named Russ Iser. And so you can see that this area, the sport fishing community actually calls this Sand Bass Junction because they know exactly what they're doing. They know that they're on the breakwall most of the year, and they'll go out to the Huntington Flats right here during the summer months. Okay, so the methodology that I chose to use was parallel transects. This is a graphical depiction. My lines did not look this clean because it's hard to drive a straight line with a boat, right? But these were basically 3 nautical miles long, and they were separated by bottom depth contours. So from 22-meter bottom depth contour to 28 meters of bottom depth. Okay, so this was my first field season when I looked at the horizontal distributions. And so I looked at the full and new moons in both June and July of 2014. And my spatial analysis, well, here, I just talked about all of this. So you can see that I actually did my transects intentionally out of order. I didn't go from 22 to 24 to 26 to 28. I actually did a closing box pattern where I went from 22 all the way out to 28, and then back up to 24, and then to 26. The reason why I did that, it's because a 3 nautical mile long transect takes the better part of an hour to complete. And I didn't want depth and time of day to serve as some sort of a statistical covariant, right? Because if I went from 22 to 24 in order, then you could potentially have variation across different times of the day that would covary with it, okay? So my statistical analysis, I don't want to get too much into statistics for the purposes of this talk, but basically I ran what's called a 2-way analysis of variance. And what I did was actually slice my transects into 10 equal replicates. And I did what's called a random stratified design, okay? So for my vertical distributions, I used the same transect pattern, except I did all 4 lunar phases across 4 seasons. I actually compiled 4 seasons worth of data. 2010 and 2012 were collected before I joined grad school, and then I collected the 2014 and 2016 data. And the data points that I used was distance from the bottom. That makes logical sense. I also actually did distance from the surface too, which is the actual depth, but distance from the bottom tells you the same basic tale here. And again, statistical analyses, I used a 1-way ANOVA because I only had a single predictor variable, and I actually used residual values. I'm not going to get too far into that, but basically I had to have a way of correcting for annual variation, which is why I did that. So I'd be happy to discuss why I did that in more detail after the talk, if anyone's interested. Okay, so shown here are my cruise tracks for my first field season, which is looking at horizontal spatial distributions. So it's separated by the full moons on the left and the new moons on the right, and the upper panels are June and the lower panels are July. So these little black dots here are actually my barred sand bass detections. And land is right here, this little sliver of green. So you can actually see that during the full moons, they're pretty evenly distributed across all the different depths, the depth contours that I looked at. But one of the compelling things that I noticed was that they appear to be limited mostly to the shallower transects during the new moon phases. And that's actually consistent between both June and July of this year. okay? So right here is a graphical depiction of that same data. And you can see, whoops, let's back up here. Okay, so this is a graphical depiction of that data. So the x-axis shows different transect depths that I drove across. The yellow bars are representative of the full moon data, and the blue bars represent the new moon data. And what you can see is that they're, once again, they're pretty evenly distributed across all depths. There's roughly the same number on average of barred sand bass at the different depths. However, during the new moons, you can see that a majority of them prefer to be more shallow, or they seemed to be more shallow, and it tapers off to very few individuals out at the deeper transects. And this is actually a significant lunar phase depth interaction. Okay, so shown here is actually sort of a modified residual plot, and this is looking at my vertical distribution data. So it's a little bit tricky to kind of understand what this is showing you. But basically, I've got my 4 lunar phases separated here on the x-axis, and the x-axis actually serves as sort of an average line. Okay, so this is residual data, and basically what the height of the bars tells you is departures from a mean. So this is 0 data. Or this x-axis right here is actually a mean line. So for instance, the full moon, they were about 0.5 meters lower than average. They're a little bit deeper than average, okay? And the third quarter moon, this is actually where my significant values are. They were roughly 2.5 meters above average. They're higher, about 2.5 meters higher than average, if that makes any sense, okay? This is actually not what a typical residual plot looks like. But in this way, you're able to actually more easily see it in a presentation, so to speak, okay? All right, so let's talk about what all of that means. So when you look at these data on horizontal spatial distributions, and you'll see that they seem to be more spread out and into deeper areas during the full moons when compared to the new moons, there's a possibility that they may be following their prey. Back in the 80s, they actually, there was a study that went out that talked about northern anchovies and how they distribute themselves based on illumination levels in the water. So when you think about a full moon, the nights are brighter, the light penetrates farther into the water. And so there's a potential that they may be their prey to the barred sand bass, right? Barred sand bass is a piscivore. Their prey may actually be existing farther out and deeper, and the predator, the barred sand bass, may simply be following their prey. When you look at the vertical distribution data, once again, the third quarter moon is where the significant value lies. This is where we believe they're actually spawning. This is when they're most active because they're increasing their vertical space usage. And it turns out that many grouper and snapper species, which are, at least groupers are very related to ceranids, they also spawn during the full to third quarter moon phases. Okay, so if you try to relate these two, there is also potential that because the full moon is directly before a third quarter moon, there is a chance that they may also be spreading themselves out. This may be indicative that they are more active during the full moon because they may be gearing up for a spawning event. And during the third quarter moon, they're a lot higher in the water column. So this actually may show a connection here. Okay, so you can't look at lunar phase without also thinking about tides as well. Okay, so going back to Phil LaBelle's idea of transport to ideal juvenile areas, I decided to look at what the tides were doing. Okay, so when you think about it, tide is when the water level goes up or down. But what's also happening is what's called a tidal current, which as the tide goes up, there's actually an overall movement towards shore. And when tide goes out, there's an overall movement going away from shore. Okay, so there's actually a bit of a tidal current here. So I decided to look at what the tide patterns look like during the different lunar phases during the summer months. And so what I've done here is separated out the periods of time where we believe that they are spawning. This is, based off the McKinsey et al. Study from 2014. So it goes from about 8 a.m. to 3 p.m. Okay, and we're going to center it around the third quarter moon because this is the significant value that I had in my vertical distribution data. So what is a tide doing? You can actually see that this period of time starts off with an outgoing tide and ends off with an incoming tide. So once again, what actually happens when the tide is on its way in is that there's overall movement of water going toward shore. Turns out where you find a lot of juvenile barred sand bass, particularly in the fall, you'll find young of the year barred sand bass that can range between guppy size to about goldfish size. Okay, they're really cute. And you actually find them in near shore environments, places like in salt marshes or within bays, in sort of protected environments. And so there is a potential that this tide cycle is promoting that transport shortly after the spawning event toward ideal juvenile areas. So obviously this is just discussing the possibilities. We don't have verification of this, but this is just could potentially be a reason why we see this pattern. All right, so what is the take-home message behind the work that I did? Well, first of all, once again, I am a big fan of fishery independent data, data that's collected independent of fisheries, right? Because you get unbiased data. You can actually use sonar for stock assessments as well. You can actually physically count fish using sonar, or actually have the program estimate the counts of, let's say, a large aggregation. It can also show you a fishery independent approach to seeing where these aggregations are in time and space. So if you take anything from this talk, is that sonar could potentially be a very valuable tool for resource management. Additionally, these sand bass aggregations can be predictable in time and space, looking at different abiotic factors, different environmental cues that determine potentially where they are in time and space. And so I believe that tightening the regulation on these fish may not necessarily be enough without some sort of a seasonal or temporal or spatial management protocol. Otherwise, they're probably going to continue to decline if people are still catching them at the numbers that they are. Okay, so in a future direction, this is actually more of a personal future direction, and that's looking at thermocline depth. So Chris Lowe from Cal State Long Beach actually approached me after my talk at one of the WSN conferences, and he had a student that figured out that barred sand bass may actually be locating themselves around thermocline depth. Okay, so thermocline is when there's a sharp decrease in temperature as you go down deeper. So my thought was, well, I don't have that information, right? I don't have the kind of high resolution data that you would need in order to relate their depths to thermocline. But him being an acoustic guru, it actually told me that I might be able to get some acoustic signal off of thermocline. Because if you think about it, let me actually show you a bit of a graphic here. So I actually did some CTD scans of one or a handful of my transects during my first field season. But again, I didn't have that high resolution data, or I didn't do it continuously. Okay. But the echogram can actually show some acoustic scatter off of that density difference, because when you have warm water up at the top and you have cold water on the bottom, there's a potential that you'll get acoustic scatter off of that density variation. And it turns out he was right. So I had about a week or so left of my software license before it expired. And I decided to align some of my CTD scans with the echograms. So you can actually see right here, this is a temperature depth profile. You can see where there's a couple of thermoclines here. There's a decrease in temperature here, and then there's another one right here. You can actually see that there's what I used to think was just noise. This is actually some acoustic scatter off of that density variation. I'll show you another one. So right here, this is actually one that's quite a bit more obvious. This is actually indicative of a stronger thermocline. If you notice right here, how well that lines up to where the thermocline actually is. And then of course, right here, you can see once again, I have thermocline data. I actually still have all these raw files, and I could at some point, if I have a spare $1,000 lying around, I could actually get this software again for a month and be able to do this analysis and actually add more to this story here by using thermocline depth, which apparently I actually have. So this is a bit of a like, again, this is a personal future direction. This is where the direction that I would like to go if I were to continue working with these data. So that's basically all I have for you. I'll entertain any questions at this time. Again, thank you all for showing up and showing interest in the work that I did. And yeah, I'll open it up to questions. Let's see, I'm going to go ahead and stop sharing. Okay, is anyone still awake?
Yeah, thank you for your talk. It was very informative for sure. The questions I had, you pretty much answered in your discussion section. So,
Okay.
so I got no questions at the moment. But if anyone else has any questions for Calvin, please go ahead and unmute.
I'm gonna stop share here so I can see. Well, I see mostly names except for Anita's face.
Hi, Colin.
Hi.
So you mentioned they to reproduce, they kind of swim up, but that only happens like once, right? So maybe this movement might not really be driven like by their reproduction. Or do you think, like, maybe
So their vertical spawning rushes actually doesn't happen just once. It happens continuously. So when they are actively spawning, what will happen is there's a massive aggregation of a bunch of males and females all together. And what will happen is the females will branch off from the group and be chased by males. Okay, and they'll go up and they'll do their spawning rush. And then, you know, there may be another group that does the same exact thing, and they'll be doing that continuously throughout this aggregation for that period of time. And so individual fish would probably only be capable of doing that once a day, only because they've released their gametes. They don't have that hydrostatic pressure within their gonads to release a second time in a given day, so they have to develop more. And this is what we believe is the reason why they go down to the bottom to rest and to feed and to try to produce more. So I guess I don't know if that answers your question, but certain individuals, yes, would only do that, you know, once a day, but collectively as an aggregation, this is happening repeatedly. I wish I had included a video of another species. It's actually some snappers. There's a really good video that shows what spawning rushes actually look like. I probably should have added that, but it's a really spectacular sight. But yeah.
Thank you.
Mhm.
And how many fish were you able to record? Like maybe like an average amount, like at the depth?
Oh geez. What was my number? Are you talking about total number overall that I got?
I don't know, just like a rough estimate, like how much fish you were able to count with the device.
Oh man, there were thousands of detections that I went through. There were a lot.
Yeah, I'm just curious.
Yeah,
curious how
there
much
was.
you could detect.
I want to say it was somewhere around like 1,400 or something like that. And yeah, there was I did a lot of all-nighters and just individually counting. Like there was actually probably a much easier way to do it, because the software itself is capable of doing a lot of things automatically. It's just in the amount of time that I had, because the software was very expensive. I didn't have a lot of time to really figure out how to do it in I guess an automatic way. I'm sure there's a way that it could be done, but this is a project that probably would have worked out better as a long-term monitoring project. In the amount of time that I had for grad school, I was not able to use it at its full potential. I wish I did, but using technology like this is a lot to take on for a master's student, I guess. You know, I figured that out. I was overly ambitious. I actually had a lot of things I wanted to do that didn't actually get done. So, but that's, you know, the typical grad school experience, I suppose. So.
No, this is really good. That's impressive. So
Thanks.
good job.
Did you collect your data like every single day for those 2 months?
No, it wasn't every single day. So for those 2 months, I actually used, this was before I used the small boats. Small boat actually cost me $200 a day to use. But before that, I actually used the RV Yellowfin, which is about an 85-foot research vessel, because at the time I was anticipating using a different boat, and there's a bit of a story behind that. I was not able to use the boat that I originally was going to, and so I ended up having to charter a research vessel to go out and do this work, and that cost about $700 a day. So I was only, for my first season, I only had a total of 4 days. So, and that's basically my June and July full and new moon periods, was actually all I was able to capture during the first season. So subsequent seasons, I was able to go out at least a couple times a week. So, and I really did try to do 3 trips per lunar phase. That was really ambitious because, you know, using that boat was $200 a day. So plus weather conditions, sometimes the swell was too high, and the SCMI staff wasn't allowing me to go out with a 17-foot boat out 9 miles offshore, you know. So there were a lot of days where I would show up and they would turn me away and say, you can't use the boat today because we can't let you out there. And so there was a lot of challenge, a lot of logistical challenges that went into using this this technology. And so I kind of wish I had a more capable boat, I guess. And so, but on average, I was going out maybe once or twice a week during 2014, 2016. So.
Yep. Thank you.
Yeah.
Were you able to share data with fisheries?
I would be able to share with, you know, like you're asking for like resource management.
Yeah, like if were you able to maybe like share your results with someone? Yeah,
So
for like management.
So I would be able to do that. There was a little bit of issues with doing that. Originally, my lab was actually collaborating with Department of Fish and Wildlife, and somewhere along the line, that collaboration had actually broken. There's there were a lot of like reasons why that was, and unfortunately, it ended up being sort of politically motivated reasons, and so that collaboration was actually broken for a lot of reasons that I don't believe should have been reasons for that. But you know, I don't want to get too far into that. But basically, it was a sort of a politically motivated reason why that collaboration was broken. It didn't involve me, but it involves some tension between my lab and resource management, and so it's the unfortunate truth behind most things that happen in this world. I try to, you know, avoid politics at all costs. I'd rather not have conflicts with anyone. And this is why, you know, this didn't involve me personally, but it was between lab and resource management. And so that's pretty much all I'm going to say about that. I kind of wish that didn't happen. I wish that we worked together, but maybe in the future, depending on what the direction that this technology goes, and the direction that, you know, the relationships between people go. I would actually like to see a collaboration, because resource management, they're in charge of regulations. And so this is something that I think, you know, should be collaborated on more than anything. So me personally, that's how I feel. So.
Yeah, of course. Yeah, hopefully someone can carry out similar project.
Yeah, I would certainly hope so. And I would certainly hope that at some point, if my name pops up and they decide to contact me, like obviously I enjoy collaborations. I enjoy working with other people. I think that's how you get a lot done. And I think there's a lot of benefits to that. So, and I used to work for Fish and Wildlife as well. So I actually know at least a couple of people there still. So I could potentially reach out to them, or no one's reached out to me yet. So I'm not even sure what the status is on that, but at this point, but something that I would hope in the future that there's a collaboration between ichthyologists from a university and resource management. I think they should work together. So.
Right. Yeah. It's needed. So hopefully it happens at some point.
Yeah, I agree. Okay.
Are there any other questions for Calvin? And I should have sent Dr. Lowe like a message and be like, hey, Dr. Lowe, you know, you're gonna be acknowledged in this presentation.
Oh
You should
yeah,
attend.
yeah. You know, gosh, like I've known Dr. Lowe for a long time, but I've known him for longer than he's known me, if that makes any sense, because I started off Cal State Long Beach in 2005, and immediately the first thing I did is I started corresponding with his grad students because I wanted experience. And so I would volunteer first chance I got, I would jump on any project I could to try to volunteer for them. And so that got me into the field. I got to know a lot of the grad students, and they were all out of his lab because his lab was looking for volunteers, and they were doing really cool stuff around stingray tracking, shovel nose guitarfish tracking, that kind of stuff. So I did a lot of I got experience in acoustic telemetry work and beach seining and stuff like that through their lab. And so, but Chris Lowe himself, to him, I was just another random undergraduate, so because of course he's way up there and I was just, like a little lowly undergrad. And it wasn't until I started showing up to conferences that he started actually noticing and recognizing my face. He's like, oh yeah, you're the one that's been volunteering for my students, and so he started to actually get to know me from there. And then of course, when I started working for Cabrillo Aquarium, my former boss there actually knows Chris Lowe and Gwen Goodman Lowe very well. Like she has dinner with them occasionally, and so I got to talk to him a lot more one-on-one during my time there because of that. So that's the reason why, like, I've known him for longer than he's known me, because when I first started as an undergraduate, he was a celebrity, right? Like he's way up here, and so I was always kind of scared to talk to him, So, but I haven't seen him in a while. So, but if he had attended this talk, it probably would have made me much more nervous. So, but he's seen my talk. He approached me before at WSN in Pasadena. Actually, is when he talked to me about the thermocline stuff. So, but if he happens to see this recorded talk at some point, he's probably going to have a lot to say to me. He's very detail oriented. So I'm kind of, I guess I'm a little uncomfortable with that, I think, but yeah, I don't know. It is what it is. That's how science is, right? Science is all about, doing a research project and presenting your findings and opening it up for criticism. That's actually the point of it. So in that way, having the scientific mindset, I should be open to that criticism, because that's quality control in science, and I firmly believe that. So
Thank you.
yeah.
Is there any other questions for Calvin? Okay, so since there isn't any more questions, I would like to thank you for doing your talk. It was very informative. I definitely enjoyed the talk. It was definitely learned a lot.
Well, I'm glad I could do that for you.
Yeah, I really enjoyed it. It's definitely something I would have never thought about, which is pretty cool. And yeah, thank you.